All right, Tess, this is dangerous. They usually don't give me a microphone because I'm typically the loudest one in the room. And I think I've already scared off our front row. So, Peter, please don't tell my boss that. But good evening, everyone. Thank you, Jamie. So kind, so kind. Round of applause for Jamie, brave soul in the front. Well, thank you all for making the time to be here today, but also for navigating the hallways of the U of A. That is usually the toughest thing, and I promise Val and Morley, we weren't locking you out on purpose. My name is Brooke Rose, and for those I haven't had the chance to meet, my pronouns are she, her, and I'm a Director of Development in the Faculty of Medicine and Dentistry, and welcome to the Game Changers in Diabetes Research event here this evening. So as a multi-generational settler, I acknowledge that we are on Treaty 6 territory, the traditional land of the Plains Cree, Stoney, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, Ojibwe, Saulteaux, and Anishinaabe, Inuit, and Cree peoples. As a non-Indigenous person, I am committed to advocating towards the Truth and Reconciliation Commission of Canada's calls to action, specifically supporting those 18 to 24, which call to improve health outcomes for First Nation, Inuit, and Métis people, which is very aligned in the work that we do here in the Faculty of Medicine and Dentistry. In relation to today's topic, I'd be remiss to not acknowledge that Indigenous peoples around the globe are disproportionately affected by diabetes. In Canada, the rates for diabetes are up 12% among First Nation individuals than the general population, making the research that we're hearing about tonight more important than ever. We are so thrilled that everyone gave up their time this evening to be here, to wear your blue, looking great. There's only so many times you get to wear a blue jumpsuit in life, so thank you. And we are so excited for the way that you have not only shown up tonight, but you have shown up to support ADI over the years and the research over the decades. We have some incredible individual supporters and champions, our Health Foundation partners, many players in the health ecosystem here, and of course the talented research who have joined us here today. I'm also really need to give a shout out to Des Lomas from Explore Edmonton, who came all the way from Ottawa for this event. So thank you, Des, for really making it a national event this evening. So it has been a very long time, nearly 5 years, since we were able to host folks here and some of the great work that's happening. And since Dr. Peter Sr. has come on board as director of the Alberta Diabetes Institute. So, I mean, those streamers have just gone by in a flash. Yeah, lots of smiles. But for those of you who may know, for more than a century, the University of Alberta and the incredibly talented researchers, many of whom are in the room tonight, have been at the forefront of diabetes research. In 1921, Dr. James Collop was a biochemist here at the University of Alberta and was actually funded by a donor to go to Ontario to work with Drs. Granting and Best, where he refined and purified insulin for clinical use. In 1989, the islet transplantation group, spearheaded by Dr. Ray Rajat, who's with us here tonight, performed the very first human islet transplant procedure in Canada. And then, of course, in 2000, Edmonton was literally put on the map with the publishing of the Edmonton Protocol by Dr. James Shapiro, who's here this evening, and ADI colleagues like Dr. Rajat and Dr. Greg Corbett, which increased islet transplantation success from 8% to 100% at the 1-year mark. And then in 2011, the clinical research unit was opened just right above us right now, and in just a few months' time, a new trial will begin with Dr. Jean Bouteau and Dr. Peter Sr. They'll actually be looking at if a single tablet can cure diabetes. And in the last few years, Dr. Shapiro and his team, like Sosha and Adish and the group that's here tonight, have been working to create new islets from stem cells derived from patients' own blood. So it's true that it feels like we're living in the future in this space and this building, and we're so excited to showcase some of it tonight. But before we get going, we did want to give just a short video to show some of the work that's been done and the history of ADI. But it's always something we like to talk about, that Teddy could climb trees, and I'm a fat boy now. So it's something that, yeah, we always like to remember. And I think so many folks in this room have their own lived experiences with type 1 diabetes, and we'll be hearing a bit more about that later this evening. So it kind of feels like I'm your MC of a wedding right now with some housekeeping items we have to go to, so bear with me. So of course, bathrooms and emergency exits just down the hall and to the left, if you need to use the bathrooms at any point this evening. But as you proceed down the hall, you'll actually see the beautiful collection of photos that was created in 2017 as a partnership with the Alberta Diabetes Foundation and the late Richard Siemens. And Richard was a talented photographer. He was a longtime friend of the university. He was actually our university photographer for 20 years. And a tireless advocate for diabetes, as he was originally diagnosed with type 1 in 1958, if my math is correct. And although we're saddened that Richard isn't able to join us here today, I do want to acknowledge that his wife Arzu is here and their daughter Danielle will be joining us. So thank you both so much for being here and representing such a special. So we may not have the incredible photography talent of Richard here today, but we do have a photo wall outside if you do want to capture some memories. Dustin will be here from Laughing Dog Photography, which we actually found out this evening was a longtime colleague and collaborator of Richard. So I guess it's always funny how small Edmonton can be. So please feel free. There's an ADI logo out there that you can take a photo with. It's definitely not a hula hoop. And of course, if you haven't already known, that the ADI logo itself has that little bit of green on the side of the international symbol for diabetes, because we do see ADI being a part of that cure, and of course, all of you folks being a part of that journey with us. So an overview of the evening, we will get going to the panelists. I will happily stop talking. For anyone who knows me well knows that's a bit difficult. But we'll get a panel conversation going with some talented folks here, different researchers. We'll then have a brief break, and then everyone on your name tag should have a tour number on there that will be able to get you going. We'll be able to see one laboratory space as well as a more clinical space, so where things like our nutrition and exercise trials are happening in the building. And at the end of the evening, you will be able to join us back here for some more food and refreshments before we say goodbye. And now finally, you may have noticed the wonderful artwork on display on your way in. And it's incredible pieces of creativity that are beyond just beautiful pieces of art. And it is actually representative of some University of Alberta and ADI researchers that have used this unique medium to share their findings from a study called the Reshape T1D study, which is focusing on uncovering the raw experiences of people living with type 1 diabetes and navigating the healthcare system in Alberta. So shameless plug, if you are interested, Jamie Boisvenu and Jasmine Magera, as well as Dr. Rose Young will, oh, hello, Dr. Young, there you are, will be also presenting next week at a really incredible presentation. So I encourage you all to attend. But I will be remiss to not say that a very special part of this is they are, once the collection is complete, going to be selling those pieces of artwork. And all of the proceeds will be going back to a new fund that was created at the ADI called Facilitating Equity for Diabetes Research. Many of our researchers actually in this building have diabetes themselves and have dedicated their time and their life to giving back to folks that they know all too well what that lived experiences is. Out of pocket, it's an incredibly significant cost, upwards of $10,000 a year that students are paying that are actually researching diabetes. So Jamie and Jasmine inspired this fund, and I really would like to acknowledge a family that's in the room tonight that helped us kickstart this funding so that folks with diabetes actually won't have to suffer the financial burden of the past. So I would like to acknowledge the Chan family, so Suzette and Stephen, who are here with their cousin Sonia, who's actually based in Toronto. And when her and Peter had a side chat, her exact words were, "This is unacceptable, how can we help?" So thank you to you both for starting such an incredible. Now to start the evening. So I will invite our panelists, so please come and join us in these very comfortable seats you're familiar with. We did not steal them from the lobby. And of course today, it's very reminiscent to talk about all these folks in the room as of course it is Giving Tuesday as well, and to acknowledge all the generosity of everyone here today. So first up, Dr. Peter Senior. He is our director of the Alberta Diabetes Institute at the U of A and will also be our facilitator this evening. He has dedicated his career to this field, focusing on type 1 diabetes, hypoglycemia, and islet transplantation. We have been very lucky to have him at the U of A for a better part of 2 decades, and I'm not sure if I was supposed to say that number out loud. From a research fellow all the way to now, the director of the Institute. So welcome, Dr. Senior. Dr. Anna Lamb is an endocrinologist and assistant professor in the Department of Medicine at the University of Alberta. She sees patients at the Cape Diabetes Clinic as part of the islet transplant program. She has an interest in developing better tools for monitoring beta cell function in patients with diabetes as well. So welcome, Dr. Lamb. Dr. Pepper, I always like when I say Dr. Pepper, it just, sit here. So Dr. Andrew Pepper is an assistant, Andrew Pepper, sorry, lived my whole life with that, the original Brooke, is a professor in the Department of Surgery at the University of Alberta and a Canada Research Chair in Cell Therapies for Diabetes. Dr. Pepper is actually a postdoc fellow in Dr. James Shapiro's lab, and we convinced him to stay, and a member of the clinical islet transplant program. And then actually he was able to start his own lab here at the U of A, where his team examines the underlying mechanism that looks at beta cell survival after transplant. So in addition, Dr. Pepper is a member of the Canadian Islet Research and Training Network and the Stem Cell Network Teaching and Education Committee. Not at all busy. Yeah, welcome, Andrew. And finally, Jasmine McGarrah is a PhD student at the University of Alberta under the supervision of Dr. Patrick McDonald. Jasmine's research focuses on unraveling the complexities of stem cell-derived beta cells and their potential in treating diabetes. She embraces a very collaborative approach to scientific inquiry, and Jasmine actively engages with folks living with type 1 diabetes to tailor her research for really meaningful outcomes. But in addition, with her own personal 15-year journey with type 1 diabetes fueling her passion, Jasmine is also a very strong advocate for the type 1 diabetes community. Her involvement with organizations like I Challenge Diabetes and Kids in Us has been pivotal in empowering, educating, and connecting with those with T1D across Canada. So welcome, Jasmine. I'll hand it over to Peter to kick things off.
Thank you. The good news is that there is no karaoke tonight. And again, before I begin, just want to thank everybody for coming this evening. It's really very gratifying to see so many people who are behind the efforts that we're expending today. But hopefully you'll get to hear a little bit more about what we're doing and why we do it, and again, have some time to answer the questions that you may have been burning in the backs of your mind. Tonight's the night to ask those questions. So I'm going to ask each of our panelists, first of all, maybe we'll start with Jasmine if we can, but tell me a little bit about who you are and what you do. We've heard a little bit from Brooke, but there's probably a bit more flavor that you could provide for us.
It's your Beyonce microphone.
Well, thanks for having me. I'm a PhD student, so I'm definitely not as seasoned as most of the panelists here, but I study stem cell-derived beta cells in Dr. McDonald's lab upstairs on the 6th floor. So basically my role is to figure out how we can make better stem cells and how we can make them act like human primary beta cells. So we have the Edmonton protocol here, but there's just not enough cells to go around for every patient that has type 1. So my role is to figure out how can we make those better and how can we put those into patients. Eventually, hopefully, Dr. Pepper can help those go into patients among everybody else in this room. So.
And in terms of your day-to-day, can you describe what maybe one of your experiments might look like?
Yeah, so day-to-day, I usually get up relatively early. My partner might not agree with that, but I'll get to the lab and I'll get some cells, typically from collaborators. I've gotten some cells from Dr. Shapiro's lab, Nadish, particularly Dr. Nurea. And then I also have collaborators all around Canada. So I have Dr. Francis Lin, who's given me his cells, and I'll isolate those cells, and then I will electrically stimulate them and read what they're telling me. After this, then I pick them up and sequence them, and then I have other collaborators, one of which, Jack Braun's here, has been looking at my cells in an RNA sequencing field. So that's usually one day.
So very much the microscopic level, looking at individual cells and if they make insulin or how well they do it, and what they resemble or behave like healthy human cells.
Yes, exactly.
That's great.
Yeah.
Can we pass on to Dr. Pepper? Tell us a little bit about what you do, and I mean, is it just the cells, or are you going at a bigger scale than that?
Sure. Well, again, my name's Andrew Pepper, assistant professor in the Department of Surgery. I have a lab on the 5th floor, which you might be able to tour today. So I'll probably make a short story long, but I really got interested in islet transplantation as a third-year undergraduate student. A friend of mine was a year ahead of me, and she was doing a research project with Dr. David White, and largely because of the success of the Edmonton protocol, I'm going to date myself, but this was in the early 2000s. They wanted to start a clinical islet program, and I saw the blood glucose traces of a mouse post-transplant, and it just completely captivated me that you could restore blood sugar control in a mouse, and once you remove the kidney where the islets were transplanted, the diabetes would be back. And that really spawned a passion to the next year pursue a graduate or a 4th-year project with Dr. David White, and that led me to grad school. That led me to Dr. Shapiro to do a wonderful postdoc for 6 years, and then in 2018 I was able to be recruited on as an assistant professor. And to get to your question of what we're interested in is still refining cell therapies for the treatment of type 1 diabetes. We know right now it's selected to about 5 to 10% of the population. So we're trying to de-risk some of the associated complications and looking at delivering localized drug delivery, and we do this in small animal models, but we also do this in large animals, and then hopefully team up with a clinical islet program and translate these findings into patient care and hopefully improve the lives of patients living with type 1 diabetes. So we're looking really at refining cell-based therapies for the treatment of T1D.
And again, so a step up from the single cell to clusters of cells into animals of different sizes, and making sure that these cells work, will do better, and will survive longer, and I guess overcome some of the challenges that we've seen in the clinic.
Yeah, and build on the lessons learned from those small molecules and those discovery science findings. So maybe we're in the middle between them.
Okay, great. Thanks, Andrew. And if I can turn to you, so, and how did you end up doing what you're doing, or what is it that you do, and where's the link with research from your point of view?
So I did my endocrine and islet transplant fellowship here, and it really is through Peter Sr. And how amazing of a clinician and researcher he is that I've come to diabetes research. And so at the time that I was a fellow, Peter was working on developing a clinical tool for measuring beta cell function in a way that we could do it in clinic, that's practical, not requiring these kind of onerous research tests. And in doing that work, we realized that we could, that kind of clinical tool would be useful broadly in type 1 diabetes, not just an islet transplant, especially in clinical trials where we're trying to preserve islet function in type 1 diabetes. And so that's where most of my research focus is now, is trying to better measure function clinically and show the clinical benefit. I think for us in the islet transplant program, and like for Andrew, where you saw with the mice, we know the difference that islets can make. For having some beta cell function there, it can make a huge difference in terms of quality of life. And what we're trying to do is find a way to quantify that in a standardized and reliable way to try to accelerate drug development.
Thank you so much, everybody. Now, we have people in our institute here from across the country, from across the world. And the question next for each of you is, how did you sort of end up here? I mean, you might have had job opportunities somewhere else, but was there something that, brought you to Edmonton or made you stay in Edmonton? I don't know, who, Andrew, do you want to go first?
Sure. I kind of gave a segue into that. Again, my short, long stories. Yeah, well, when I was doing graduate school, I was also, my supervisor at the time was looking at developing a biotech company that looked at a place to put these cells underneath the skin. And we were able to do some large animal work. And through a conference, Dr. Shapiro teamed up with David White and wanted to do a first in clinical trial here in Edmonton, being the epicenter of cell therapies for the treatment of type 1 diabetes. And I was very humbled to be recruited to join him as a postdoc and kind of help with those trials. And it was a no-brainer, because when you're a graduate student, you're in cell therapies, do you go to Boston or California? No, Edmonton, Alberta is literally the world leader in the cell therapy. So it was an easy move here. And to be in an edifice dedicated to all lenses and flavors of diabetes has been a wonderful experience, because collectively we have different perspectives at looking at diabetes, whether it be type 1 or type 2, and we can leverage lessons learned to hopefully all together make a difference in all patients living with diabetes. So it was very easy to stay here, to come here and stay here, and I don't mind the winters at all. I like to ski and stuff, so it's been wonderful.
Sure, I guess. Yeah, so I was born and raised here. Born and raised Edmonton Oilers fan, even though sometimes they're a little questionable. But I think I spent my whole life trying to get out of Edmonton. And through my undergraduate, I did an undergraduate in pharmacology, and I took a course with Dr. Patrick McDonald, and I realized how many giants that there are here. So I didn't realize that growing up, I guess. When you're young, you always want to see the world. And then I kind of got convinced from another PI that I was working with, Dr. Harley Crata, to work on the thing that I'm passionate about type 1. So I said, okay, I'm going to do a master's, and that's it. I'm going to leave. And then it just got wrapped up in it, and it was so many amazing collaborations here that I was like, I can't leave now. So I switched over to a PhD, and I've been in Edmonton, I guess, forever. And I'm happy about that. So, yeah.
So I'm also an Edmontonian through and through. I was born and raised here. I did go to Calgary and Winnipeg, but found my way back. And so I became a part of the ADI as a fellow again through clinical trials. So both in type 1, type 2, looking at newest insulins that are coming to market, and also as a part of the islet transplant program, the first in human stem cell derived islets trials, which is very exciting. And I think what's been really gratifying or satisfying is the collaborations within the ADI that I've been a part of. I have fortunately been funded through the ADI for a pilot study looking at the effect of exercise on glucose in women with postmenopausal women with type 1 diabetes. And where that has been, I think, especially gratifying is in my clinical practice, day to day, I'm having this conversation with people about how to manage insulin and exercise. And it really is a gap in our evidence or knowledge. But I get the chance to say, hey, we don't have good evidence, but we're working on it at the ADI. And if you're interested, you can take part in that. And even though we're doing clinics virtually, like Jess or Reed, they're going to be on those calls. And if patients are interested, they have the opportunity to sign up. So it's been very satisfying that way.
Okay, that's great. So a couple more questions from me, and then we will open it up. But Andrew, I'm going to kind of tell a little bit of a story about kind of maybe how and why support is important. But I'm guessing that you didn't pursue research for fame and fortune. What was it like being a grad student? And again, I think you've already told us how long that it took you, but were there kind of studentships or scholarships that kind of moved you along your career?
Right. So you're absolutely correct that a stipend of a graduate student, a postdoc, and salary of assistant professor is certainly not lucrative. But it does provide a vocation to scratch a curiosity itch that I'll probably always have and help refine cell therapies. But starting in grad school, I went to Western Ontario, so I'm not from Edmonton, but it was a wonderful experience to work in an environment where it was that the end goal was to always try to get therapies to patients, and that was really nice to have this macro perspective on what we were doing in the lab, and some fellowships along the way were extremely important. I had a MyTex fellowship that linked with industry to help support work, and that helped me get to Edmonton as well. And then through Dr. Shapiro and a postdoc, I had some fellowships through Alberta Innovates, and then to be recruited here, it was largely due to the Alberta Diabetes Foundation that provided support to recruit for salary support and startup funds to stay here in Edmonton. So throughout my whole academic career, donation support and both provincial and federal as well, have been instrumental in continuing to scratch an itch of curiosity and pursuing an academic career, despite the lack of fame and definitely lack of fortune.
We will be selling lottery tickets afterwards, perhaps.
Maybe we could pass the hat later on.
If I could maybe turn to Jasmine, I think Brooke mentioned that you lived with diabetes, and I think my question is maybe quite blunt in some ways, but have finances been an issue for you as a grad student with diabetes?
Yeah, definitely. I think finances are an issue for all grad students, but unfortunately when you live with type 1 diabetes, you do have a lot of extra costs. Those costs can be medical technologies like sensors, it could be insulin, because sometimes insulin isn't covered. There's a lot, and I definitely was finding that through when I started my degree, through my master's, there was a moment where I was like, I don't think I can continue this journey because I can't afford it. And I know my friends and people who are around me, they suffered from me being very cheap and being very conscientious about my spending. But even with that, I could not get my head above water, and I got really lucky with a lot of studentships and scholarships that I received. So I'm happy to say I'm doing a little bit better now, but I'm hoping that we can pass that on to other graduate students that do live with type 1 diabetes.
And so I think, again, for people in the audience, the lots of charitable bodies, the Diabetes Institute, we award graduate studentships to kind of give students a stipend so they can get their degrees, but they're actually the ones doing the work in the lab to a large extent. And so it's a kind of both a research and training and education, they very much go hand in hand, but the kind of the money that comes into the Institute allows us to give the graduate studentships, which then mean that you're more competitive for national competitions. And I think we need to congratulate you on getting a Vanier scholarship this year, if I'm not mistaken.
Couldn't have done it with everybody's help and support.
Anna, if I can come to you. So you see a lot of people living with diabetes, and you mentioned this already in terms of seeing people in clinic and saying, hey, would you like to help us answer some questions? But for people who maybe haven't thought so much about research, are there different kinds of research? Should people be scared about taking part in studies, or is it something that is a good opportunity?
Yeah, so firstly, study enrollment is probably one of the major barriers for us to complete clinical trials or studies. We need to get enough participants to ensure that we have reliable studies and study results. And in terms of the studies that people can be involved in, it doesn't have to be a clinical trial where you're taking a study drug, and you need blood draws. It could be something like an exercise study like Norm Boulay at the ADI, or it could be a nutrition study, it could be a survey or interviewer study, and it doesn't have to be in person, it could be online, like with Jamie's Reshape T1D. So there's a lot of different opportunities, and I think we should be doing a better job of giving those opportunities to patients, because I think they do want to be involved and hear about what we're doing. So, yeah.
One of my colleagues said there's two kinds of research. There's talky research and there's pokey research. So again, it doesn't have to be quite as scary as you might think. And I think certainly my experience has been that the ability to contribute to give back has been something that's been a very, very strong motivator for patients living with diabetes. They know that they may or may not get benefit from participating, but they hope that we learn lessons which will benefit others for the future. Okay, and so maybe one last quick one before we open it up. So the suspense is building, I'm sure. This is a kind of a big institution. We've got lots of buildings, lots of floors, lots of expertise. I think we've alluded to that already, but I don't know if there are either people or personalities or people from other countries or different disciplines that you've had a chance to interact with that is kind of would speak to the sort of the value and the unique opportunities that we have here that you might not have had if you'd been either working or researching or studying in perhaps a different city or a different institution. I don't know. Anna, do you want to begin?
So yeah, definitely through the ADI. If it weren't for the ADI, I wouldn't have gotten into that exercise research in type 1 diabetes. It's really through Jane Yardley, who has just recently moved to Montreal, but I wouldn't have had the expertise or skill to do that on my own. And so really being a part of an institute that can bring those different skill sets together has made a difference. I can also think of John Usher, who looks at diabetes in a completely different way than I do. He works in mice and looks at obesity and diabetes, and he's done work with computer modeling and trying to predict drugs that can fix diabetes that we don't think of at all. So he's figured out that maybe this old antipsychotic drug, a drug we use in schizophrenia, we might repurpose for diabetes, and he's proven that in mice, and we're trying to work together to do that in humans. And I wouldn't be able to do that if it were not for the ADI.
Yeah, I can think most recently of a wonderful opportunity that sprung from just being in a building with individuals looking at different facets of diabetes, and one again was with exercise. And I never thought in a million years I'd be exercising rats before a transplant, but I had a conversation with Dr. Yardley and looking at could exercise, prescribed exercise, whether it's aerobic exercise or resistance training, help post-transplant outcomes, because we need about 2 pancreases per recipient. So we applied for an ADI Catalyst grant, which brings different investigators from different backgrounds together for a single project. And so right now we're exercising rats before and after transplant. It has some really interesting outcomes on our single donor success rates in rats, so we can get away with less islets by just exercising about a month prior to transplant. So it's been a really interesting scientific question that I never would have thought of if it wasn't for bumping into people in the hall and learning from their research, both clinically and in different models. And this is also tethered in with Dr. Usher as well, who's looking at energy metabolism post-exercise. So why do we see better transplant function pre and post-exercise? Looking at how the body compensates for that. And another investigator in the ADI, Carolyn Richard, looking at maybe we're changing the immune system a bit as well by exercising. So multiple different lenses looking at, again, at the same problem and collaborating together in this building. And hopefully it can lead to maybe clinical trials with Anna and Peter and Norm to exercise patients instead of rats.
And of course, the question that we all want to know is, how do you exercise rats, and do they need to wear Lycra?
Well, if you might have a tour in my lab. You could see one of the ladders we have, but we have ladders and wheels. They actually really like it quite a bit, more so probably than patients, clinical patients.
And so, Jasmine, you've also had the chance to kind of, I think, collaborate with people inside the institution and around the world. I don't know if there's any particular stories or things which kind of really spring to mind.
So I think the story that always comes to mind is often a lot of research is done outside of the lab. So when you travel for conferences and you have a few drinks with folks, often those are when the ideas come out, and kind of the unfiltered, crazy ideas. And we had quite a few, I guess, I don't know how much I can say, but we had a lot of collaborations with people from UBC about, I don't want to give too much up, but types of sequencing that we can do in these cells. So we brought it back here, and in my lab, actually, I started our sequencing pipeline from scratch, but we left functionality channels open. So as technology gets better, as AI gets better, we can actually incorporate that with our technology and our pipeline, which we've built. And also this technology was built with kind of a cross-campus collaboration, because the ADI is really good with collaborating across different institutes. So I did an EM study, or electromagnetic study, in engineering, and then I went to CSIS, which is an interdisciplinary building, and they helped me.
Not the spy agency.
No, not that
Or
one.
you wouldn't tell us about that if it was, would you?
Sorry, it's an NDA.
All right, that's great. Well, listen, I don't know, are there any questions that the audience would like to ask? We had some submitted in advance, and we can try and tackle some of those. Anybody feeling brave or?
Oh, Dr. Rajat. I don't know. Oh, here, we'll give you.
Funding is always a problem, and what areas of funding do you think are the most need to move the field forward? I mean, I think of graduate students, postdocs, et cetera, but what area do you think we should be funding to?
I think if I speak selfishly as a kind of the director, we are working hard to communicate a bit more clearly what it is the ADI wants to do. And we've got our pillars under this CURE acronym. So cure-based research, understanding the disease, building the runway for clinical trials, and educating the next generation. And if people give money to the ADI, I can help plug the gaps because, you know, it's a bit like harvest. You know, some years there's a feast and some years a famine. And so if we've got funding coming through one channel to support our grad students, but we've got a gap somewhere else, well, we can plug that gap. And I think also the ability to kind of, you know, kickstart some things and make some investments that will help everybody later on. And so one thing that's top of mind for me at the moment is we're thinking about building a national platform to kind of, it's a bit like matchmaking or Tinder to link people with diabetes with clinical trials. Because again, we talked about recruitment being so hard, and yet people don't know of opportunities. How can we bridge that gap? So we have been working quite hard to think about how we can build that runway so that if we do invent a new treatment or a new therapy, how do we get the trials done? It would be a shame if they had to be conducted in other parts of the world. So I think grad students is kind of our bread and butter. I think this Catalyst grant that Andrew talked about, that's been a really key thing. And again, thanks to the Blanche family for your support in investing in that, because I think it's really helped bring groups together. Complex problem, nobody's going to fix it on their own. And so bringing groups together, and I think we've heard today about those themes of, well, this person helps me and they've got this insight, and, you know, Jasmine evaluates the cells that James is growing that then will be transplanted into the clinic. So we're also very interdependent, and building those relationships, making that easier, is what I would like to help invest our money in.
Any other questions from the group? Nothing. Oh, yeah, Steph.
I'm just curious with AI and how it goes into your research. I know AI is throughout a lot of different industries, but I'm really curious how you use AI in diabetes research.
Thank you.
That was really subtle. Yeah, yeah, really.
Yeah, so AI has been kind of our talk for the last 10-ish years, I want to say. And now we're seeing a lot of connections to research. So we can predict how well, so my lab has worked on predicting how well beta cells will function based on certain parameters like expression of genes, for example. So if, let's say, we can expand this to stem cells, a whole bunch of groups can create a whole bunch of stem cells. We can run it through this algorithm using our parameters, so insulin secretion or gene expression or any other parameter that we are interested in at the time. We can spit out a score, and we can say, this is 50% like a beta cell. And so that AI is speeding up what we're finding. We're also finding predictive algorithms are helping a lot with our RNA sequencing specifically to tell us this is what we think is going to be the part that you should target. So in lab, then we can go target that pathway and hopefully create a little bit of a stem cell that's better and closer to human primary cells. How's that?
Also, there's an opportunity now with hospital records or electronic medical records for diabetes management in hospital. We have patient characteristics, the medications that they're on before coming into hospital, have a sense of what their control was before with their blood work, what their glucose levels are in hospital, what insulin they're receiving. We can use all that data, put it into an algorithm, and try to model glucose and glucose response to insulin, and hopefully help people so we're not needing necessarily diabetes specialists to manage diabetes in hospital, but offload some of that work with the machine learning.
And that would hopefully make things safer, because hospitals are often not a particularly safe place for people who live with diabetes, unfortunately.
Oh, I was just going to add, and we also just placed last year the first Alberta Health Services Chair in Artificial Intelligence. So who's working with Alberta Health Services to access data to do some of the information in other disease groups. So again, tapping into that bigger picture too across the university. Maybe one time for one last question before we have a break. Any burning questions?
Well, one of the online questions was asking about kind of type 2 diabetes, because I think a lot of what you've heard tonight has been, you know, about cell therapies and things like this. So we'd be back to that you of the cure thing. So understand the disease. So the work that Jasmine is doing, you know, she knows what a healthy beta cell looks like and should do. And that's highly relevant for type 2 diabetes, because really we don't fully understand what causes type 2 diabetes. It's easy to say inactivity, poor diet, overweight, but those are merely the triggers. Like, it's the genetics underlying that is the problem. So a lot of what we are doing that, you know, might at first glance seem, you know, oh, that's a cell replacement treatment. Well, actually, it will have spillover effects. And I think probably the majority of the work that goes on in Pat McDonald's lab, he supplies islets from healthy donors and people with diabetes to labs across the world. And again, probably the vast majority of them are looking at drug targets for type 2 diabetes. And I think, you know, both the nutrition and the exercise group, I mean, Norm is working, I think, with Rose on a sort of an intervention to reverse and regress, cause remission of type 2 diabetes. So on the one hand, we're not a one-trick pony. We do have some clear focuses. We certainly have expertise, and really what I think our strength is in bringing all of these strands together to really lean in together to move forward on this, you know, hugely important disease and condition that affects so many people, many of whom are, you know, known to people in this room. So it's for your friends, for your relatives, for my friends, for my relatives who live with diabetes. That's why we do this work. And I want to thank you all for the huge contributions you make as supporters of all that we get to do in our day jobs.
Awesome. Well, please join me in thanking our lovely panel guests for this evening. And now the fun show and tell of the evening. Oh, thank you. I have 2 microphones, so that's dangerous for everybody. Well, I just wanted to also give a shout out to our wonderful trainee working group who stepped up to help support a lot of the tours happening today. So thank you so much for those folks to be a part of it. So on your lanyard, you will have a number this evening that you'll be able to part of, so either with our tour group 1 through 4. So if you are with tour group 1, you'll have the wonderful Zosha, who hand up there at the back. So we'll make our way out to the lobby and you'll be able to go to your tour leads, and you'll be able to see Dr. Shapiro's lab as well as the Health Nutrition Research Unit and Clinical Research Unit. Tour group 2 will be with Dr. Senior and myself and Vince Rogers, the Operations Director of ADI, and we'll head upstairs to the Health Nutrition Research Unit before swapping with Zosha's team. Tour group 3 will be with Nuria out the back here as well, and who is a postdoc fellow in the Corbett lab, and we'll be taking through Dr. Pepper's lab and then going to the Physical Activity Diabetes lab. So there are no rats there on ladders, Norm? Okay, great. And Dr. Norman Boulay and his team, so Justin Reed as well, will be taking you folks through the Physical Activity Diabetes Lab, and then we'll do a bit of a swap over. So we'll probably have a break for about the next 7 to 10 minutes to hit the bathrooms, grab refills of water, and we'll meet you kind of in front of the elevators, and we'll portion off. You'll see lovely signs. We don't have the umbrellas like the tour guides, but we do have signs. And just before we go, we have one last announcement. So everyone had put their name in a draw. It's a very exciting prize. You get to have lunch or coffee with Dr. Senior and get to pick his brain on all things diabetes research. So, I don't feel like I should pick. Let's see here. Angelina, would you like to draw the name there? So, do we need a drum roll? Like, I don't know. Is that not? I feel like you need a big chicken car. Oh, it's there. Oh, Paul Sorensen. There you are, Paul. There you are, Paul. So we will touch base, and you'll have a chance to pick his brain here. So, well, thank you all very much. I know the event is Game Changers in Diabetes Research, but we truly believe you are all the game changers that are making these things happen, from some of the examples we used today, but just being here this evening. So enjoy the rest of the evening. We'll see you out in the main foyer, and we'll be able to get you on your tour groups and off and running. So thank you so much again.